thank you so much for having me. You've been incredibly gracious hosts so far. It's been an absolute pleasure to talk to every one of you. And I'm really looking forward to reading those papers coming out of your PhD. Right, so this is my lab last winter. I promise these are not Halloween ghosts. These are people actually have legs. These are just some new additions to my lab. What you cannot see on this picture are all the yeast cells that are lurking at us from behind from this ancient oak tree. We do know that they're there because we have recently started collecting and isolating yeast from oak bark samples all around Sweden. So first, as I promised, let me give you a quick rundown of my kind of buggy and fishy academic background. So I did work on fruit flies in the past. This was an undergraduate project where I was put to work to measure wing sizes in many flies. But I did my PhD on African cichlid fish with Ole Seehausen in Bern in Switzerland. I did my first postdoc on trout, where I looked at quantitative genetics and maternal and paternal inheritance patterns. And I only changed model systems to yeast quite late in my career, actually. So this was about 6, 7, 8 years ago now, when I met Mike Brockhurst, and he said to me, hey, what you do is really cool with these fish, but why don't you do that in yeast? It's much easier. And he convinced me, and I actually never looked back. I think it was a really good thing for me to do. But what I wanted to point out here is that I did actually do 7 international, well, European, but international moves over the course of my career. And this kind of life comes with some costs, right? I did talk to some of you today about good strategies for your academic future. And I do generally recommend that either you stay with the same model system, but you change your question, or you do the opposite. And this is what I did. So I changed model systems, but I kind of remained faithful to the same scientific question, which is, what is the role of genetic variation in adaptive evolution? But the costs are also quite severe if you want a career in science. So this is a sad truth that 12 years after their PhD, only 44% of female tenured faculty have a partner and children versus 70% of male tenured faculty. Whether this is one of your life goals to have a partner and children, I don't know, but this just points out that there are definitely inequalities in our field. And you can see these costs in my own publication record. I call these environmental effects on my publication record. So every time I had a baby, you see a dip in my publication output. A large dip actually also came when I got a tenure track position in Sweden, in Stockholm. Because you have to build a lab, you have to hire people, you have to become a project manager, you have to manage your own budget, and those things take a lot of time, teaching as well. So all these things are costs that you have to pay, and I think it's really important to see the people behind these careers as well. I'm happy to discuss work-life balance things with any of you. You're welcome to contact me, and we can talk through this. But back to the science. So my lab, it's a yeast evolution and yeast genomics lab, and we're interested in what happens when genomes merge, when they recombine, and when they diverge again, and what happens with this genetic variation that's contained in these populations and how they can perhaps contribute to populations coping with environmental change. So, meant to say, do you guys recognize this little drawing that I painted on a petri dish with yeast? Anyone shout out loud? That's right. So that's right. So this is the first ever tree of life that Charles Darwin drew in 1837. And a lot has happened since, right? So the field of population genetics emerged. And then shortly after, the field of yeast genetics emerged as well. But other than some convergence in shirt choice, there are actually some incompatibilities between these two fields. So while a population geneticist studies lots of interesting organisms, allows for lots of sex, at least in study organisms, and uses experiments and models to see how things work. They normally leave their study organism at work. You don't take your flies home with you, I assume. Yeast geneticists think there is really just one interesting organism. They like to control everything. They don't normally allow for sex. They break things, so genes most of the time, to see how things work. And they often take their study system home to make beer, actually. So we still have to brew a Stelkens lab beer. We haven't yet, but I have many requests. So I kind of think of my own lab as this kind of hybrid version of these two worlds. So I try to combine the best and the worst things from both of these fields. So here are some good reasons to use yeast and not fruit flies for studying evolution. So first of all, I can, which is probably the biggest difference, I can freeze my populations for a long time at -80, and I can pull them back out of the freezer and I can bring them back to life. So I kind of have this fossil record in my freezer, but these are kind of living fossils. So I can streak them out on a petri dish and they will come back to life. So that, for long-term evolution experiments, that's really nice because I can just pull out the ancestor from the freezer and I can compete them together in media, liquid media, with an evolved population to see how their fitness changed over time. I can also use both liquid and solid media. So that means that I can look at entire populations at once, but I can also streak them out on solid media, so I can look at individual genotypes. We also have a lot of great molecular genetics tools at our fingertips. So we recently, I know I'm late to the game, but we just established CRISPR-Cas in my lab. So this is really neat. There are more reasons why yeast is a good system. One is that we can really control the mode of reproduction that we want. So yeast is normally a diploid. So we start the cycle here. Sorry, there's a bit of delay in this pointer. So this is normally how yeast exists in the wild. It's a diploid, just like you and I, and it just reproduces asexually by budding off cells. When you take all the food away, when you starve yeast, they will go into sporulation. That means they undergo meiosis. And this is a run-of-the-mill meiosis with crossing overs, recombination, and independent segregation of chromosomes into the daughter cells. It makes these 4 haploid spores, and these haploid spores can either just bud off asexually, they can make more clones of themselves in a haploid stage, or they can find a cell of the opposite mating type, and they can mate and become a diploid again, and then the whole thing starts again. This is very handy if you are interested in hybridization, because you can actually cross very divergent species of yeast together and make F1 hybrids and sporulate them and then get recombined F2 hybrids very easily. This is a little bit of a preview of tonight, perhaps. So do you guys like lager beer, ice cold lager beer, or are you more the IPA types? I don't know. Anyway, so I don't know how many of you know this, but every lager beer that we produce and drink is the product of a hybridization event in the past between 2 very, very divergent species of yeast. So this happened like maybe 500 years ago in the basement of a maybe German or maybe Austrian monk, where these 2 yeast species got together, they hybridized and made kind of like an industrial hybrid species that we call Saccharomyces pastorianus. And this hybrid strain combines the fermentation capacities of the brewing yeast, Saccharomyces cerevisiae, with the cold adaptation of this wild species, Saccharomyces eubayanus. So we started being able to make cold fermented beers that would last longer in the basement. Okay, so if you already like lager, beers, maybe you appreciate it even more because here you can see the most recent phylogeny of all the 8 Saccharomyces species that we know of. Stopped working now. All right, I'll try to do without. So these are the 8 known Saccharomyces species that we know of. I'm sure there are more, but we haven't found them yet. The 2 species that you see there, Eubayanus and Cerevisiae, are the ones that have hybridized in industrial environments to make lager beer. And these guys are almost 20 million years divergent. So these are really, really divergent species. There are some that are a bit more closely related, but these divergence times are about as different as rhesus monkeys and humans. And you can still get viable hybrids from them. So there's a huge amount of genetic variation in the system. But not just that, there's also a huge amount of ecological diversity in the system. So we have isolated yeast from woodland habitats, from fruit, from insects, from plants, soil, vaginas, the bloodstream of patients in hospitals. So they can live in many, many different habitats. And also, of course, in the usual kind of industrial fermentation environments. Great, but probably the most interesting aspect for us is that we can use them for experimental evolution. So the idea is very simple. You're all familiar with that, I think. Often you start out with a clonal population. You see mutations pop up over time, and then you can see them go to fixation or not, and you can measure their fitness effects, etc. There's a really great recent review article on this, if you're interested, by Deepa Agashi et al. And AMNAT, which kind of revisits the role of genetic variation and adaptation. So in my lab, we like to start this process, but from different time points. Sometimes we use clonal populations. Sometimes we use populations that have a little bit of standing genetic variation in them. And sometimes we use very, very diverse populations, which are hybrid populations. So I want to tell you 3 different stories today. I'm going to start with the leftmost story about interspecific hybrids. But I also have one that used intraspecific crosses and one that starts with clonal populations. So with these interspecific hybrid populations, we wanted to know whether hybridization can contribute to adaptation. This is something that has been shown in different systems. Something that we don't know and understand very well as a field yet is under what genetic and ecological conditions hybrids can actually be adaptive and successful. But of course, there's a huge problem with hybrids is that most of the time they're terribly unfit and they cannot reproduce. They're sterile, right? And yet yeast hybrids exist and they thrive in both natural environments and industrial environments. So we have evidence that almost every species of yeast has hybridized in the past. I told you about the lager beer before, but hybrid strains are also used in the winemaking and cider making industry. But there's also work showing that they hybridize in natural environments, forests, and fruit. So my postdoc, Devin, who's moved on to University Hospital in Edinburgh by now, he wanted to know whether the genetic divergence of the parents actually predicts hybrid viability, right? This is a kind of a classic experiment that many people have done also in fruit flies before. So he measured hybrid viability across the entire genus of Saccharomyces. He used some published cross data and made some of his own crosses as well. And this is what he saw. So not very surprisingly, so you've got genetic distance between species at the bottom, and you've got hybrid spore viability here on the y-axis. So this is a measure of reproductive isolation, right? And you see that reproductive isolation increases with increasing genetic distance between species. One interesting detail here, where a lot of genetics research is going into at the moment, is that these hybrids, especially at large genetic distances, are often highly aneuploid. So they have too many or too few chromosomes. They don't have your typical 32 chromosomes of a diploid yeast species. And this is caused by a mechanism called anti-recombination. So anti-recombination, so if you have 2 very divergent yeast species and they're trying to line up during F1 hybrid meiosis, they cannot find their homologous chromosomes. So that means there is no crossing over forming, and that means that chromosomes missegregate into the hybrid offspring. So you often get gametes that have too few or too many chromosomes in them, and that is often lethal. Strangely, structural variation and also these classic Dobzhansky-Müller incompatibilities don't seem to play a very big role in speciation in yeast. Okay, so we can think of this genetic variation that's brought about by hybridization as this kind of sweet spot between perhaps inbreeding and outbreeding depression. This is genetic distance between the parents and hybrid fitness on the y-axis again. And there are, certain things that probably play a role. So at that high hybrid fitness, maybe you get a lot of kind of intermediate phenotypes, but they're still viable and fertile. And then the further out you go, the more novel, transgressive, and probably also aneuploid phenotypes you get in the hybrids. But what we know very little about is what role the environment actually plays. So if you have a transgressive hybrid that might be really bad in the two parental habitats, but it might be really adaptive and useful in a novel habitat. So what we often do in my lab is we take two different species of yeast, we make F1 hybrids, send them through meiosis, and have these recombine F2 hybrids. Not many of them survive, right? Only about 1 to 5% of these spores actually make it, but then you have a stable hybrid genome. And we throw a lot of trouble at them, right? So this is one of our favorite treatments. We call that the hangover treatment. So copious amounts of alcohol followed by caffeine and aspirin. You can stress yeast cells with pretty much the same stuff as you can stress human cells with. Just to see, in what kind of ecological context, environmental context, some of these F2 hybrids are successful. This is an example from salt, where we just threw tons of salt at these populations. So these are 96-well plates, and we grow them on these 96-well plates in liquid media. Every well has a different population in it. There were exactly 1,300 populations. These are F2 hybrids. We also tested F1 hybrids and the two parental species. We also made some intra-specific crosses to see how well they are doing in these novel or stressful environments. And we transferred them every 3 days to another plate that had even more salt in them, right? And this went on for about 27 days until everybody was dead. So what we wanted to see is the population survival in these 5 groups. And these are the days in deteriorating environments. So we will have in red the F1 and F2 hybrids. We will have in black the F1 and F2 within species crosses. And then we'll have also the data on the parents and how long they were actually able to survive in these environments. This is the parental data. This is averaged across the 2 species. And you see that after about 15, 18 days, everything was dead. That's how long they held out in these stressful environments. Compared to that, the intraspecific crosses, so they had a bit more genetic variation in them, they made it a little bit longer in these pretty nasty environments. But the actual interspecific hybrids survived, at least some of them, for a much longer time. So this is only 6 days, but if you imagine that these cells reproduce every 2.5 hours, this is asexual reproduction only, that translates into millions of cells more than the parents. And this was cool, right? This was interesting. So that massive amount of new genetic variation that is created through hybridization can actually be adaptive in a stressful environment. And what's also interesting here is that the F2 hybrids are doing actually a little bit better than the F1 hybrids. That means that it's not just due to heterozygous advantage, not just heterosis, but due to the recombination of divergent genetic elements here, meiosis. So, okay, so that's cool, but what's the genetic basis of this? Of course, that's the next question, right? So Devin asked again, can we predict hybrid fitness from the ecological or genetic features of the parents? This would be pretty useful, right? If you think of agricultural breeding, if you think of climate change research or conservation biology, it would be really handy to know from the genetic and ecological features of the parents how fit their hybrid offspring will be. That we can make more food that way. Or maybe if you're a conservation manager and you're trying to protect an endangered population, you might want to decide whether you want to allow for supportive breeding from a slightly different population or not. So Devin asked if there are any, so there's lots of published yeast hybrid genomes online. This was a kind of a pandemic project because we couldn't actually go in the lab and do our experiments. So Devin looked for all these data online, and he did comparative genomics on 200 plus hybrid genomes, and he mapped them synchronously to all 8 Saccharomyces species using read depth and K-mer analysis. So they came from a large variation of different ecological backgrounds. So first, this is the first kind of result that from this analysis, that parent ecology doesn't seem to predict hybrid viability very well, or hybrid success, I should say, very well. So naively, you might think that a population, is that working again? No. So naively, you might think that a species that is specialized on soil, right? So the third one from the top there, is making successful hybrid crosses. Aha, that one. Oh, yes. So perhaps you'd think that a species from soil might make very good offspring with other species that were isolated from soil, right? But that's not the case. So the expectation, if this was true, that ecology predicts hybrid success, you'd see a lot of blue dots, blue large dots here on that line, on that diagonal, but that's not the case. And what we think happens here is that, so some of these domesticated strains from fermentation environments, but also lab environments, are probably distorting this picture quite a lot. And we think that's likely because domesticated strains have a lot of loss of function mutations. And when you cross it to a wild species, genomic background, they can be, these deleterious mutations can be rescued by these wild alleles. So this is due to simple complementation leading to heterosis. Anyway, let's look at the genomic patterns that we found in these crosses. So we did find strong chromosome by species interactions in the composition of these genomes. So this is one particular cross, Cerevisiae with Eubayanus, and you'll see about 80 or 100 hybrid genomes as rows here on that plot. These are the 16 chromosomes of yeast, and these are the hybrid genomes mapped to both of the parents. In blue, you'll see the one species, and in red, you'll see the other species. And so there were some patterns here that you can see by eye, which is that, for instance, chromosome 6, all across these different hybrid genomes were more likely to come from Saccharomyces cerevisiae, whether chromosome 9 was more likely to come from the other species. So there are some things going on in here, and this is just one of the crosses, and we have 6 other crosses where you see similar patterns. So there are definitely strong interactions between chromosomes and cross. Okay, one other interesting result from this analysis is that more genetically distant species here produce more similar hybrid genomes. So this is a little bit counterintuitive, perhaps, at first, right? Because you'd think that 2 very divergent genomes might make, might have more random hybrid genomic outcomes, because, there's a lot of havoc going on here. But the opposite was the case. So the more divergent 2 species are, the more similar are the hybrid genomes that they produce. And we think this is likely due to stronger negative epistatic effects at larger genomic divergences that puts tighter constraints on these hybridization outcomes. So there is a limited number of possible solutions how you can build a successful hybrid genome. This is the last result from that particular analysis. So we also found that aneuploidy, which I mentioned before, is really, really common in hybrids. These are the the 6 different crosses here in the rows, and these are the chromosomes again. And we found that this was highly cross-specific. And there's effects like, for instance, here, chromosome 5, across all the genomes that we looked at, chromosome 5 in this particular cross was more likely to be there in several copy numbers, not the euploid copy number. The opposite was the case for this species here, where you see a chromosome loss on chromosome 13. So instead of showing you conclusions here, I'm going to take you a little bit on this kind of thought experiment about aneuploidy. So aneuploidy clearly is a meiotic error, right? A segregation error that messes up genomes in hybrid meiosis. But is this a bug, or is that a feature of hybrid genomes? So we know that aneuploidy can disrupt homeostasis, cell homeostasis. You know, different dosage effects are running wild. It can lead to cell death. We know that it causes genetic disorders and cancer. But on the other hand, aneuploidy can also provide adaptation to environmental stress. We know that in Candida, for instance, it can cause resistance to antifungal drugs. It can also allow for transitions from commensal lifestyles to pathogenic lifestyles. So we know, for instance, that Cryptococcus can infect humans, and in the moment of infection, they actually become aneuploid. And it can enable the colonization of new environments. And this has been shown in yeast. This is not our data. So this is adapted from Jona et al., where they've seen that this was a kind of a long-term evolution experiment over several hundred generations. Exposing populations to heat stress leads to aneuploidy-assisted adaptation. So the population gains one extra copy of chromosome 3, which it then loses again later. And the reason for that is probably some more elegant solutions pop up. So some compensatory mutations in the rest of the genome, and then that aneuploidy is shed again from the genome. So it's a very kind of unstable route to adaptation, but it can help in the short term. We also see that popping up in our own evolution experiments. So here's an example where we found this was a population exposed to higher amounts of ethanol for 1,000 generations. And we saw that chromosome 10 evolved an aneuploidy that was really stable over time and stayed there for a long time, which suggests that it's adaptive. So I just am thinking about what this could mean. So I think it could mean that this kind of coincidence of hybridization causing lots of aneuploidy, being exposed to environmental stress, could give hybrids a selective advantage, which could explain why we find them quite often in natural but also industrial habitats. So these kind of, what starts out as a meiotic error may actually cause these long flights across the fitness landscapes in hybrids. Okay, so this is the second story. So we talked about hybrid, yeah, absolutely. So no, because the way that we determined genomic similarity is the number of shared k-mers, so shared windows along the genome. So there were blocks of the genome that were identical, and that was more often the case the more divergent the species are. Aneuploidy is just a copy, right? An identical copy of a chromosome that was already there. So, aha, so you're suggesting, okay, that is a very good question. And I have to say that I don't know the answer to that right now. I do hope very much that we have standardized for copy number variation there for that analysis. Yeah, but that's a great question. I'm gonna have to dig into that. Yeah, thanks. I forgot to say you're welcome to ask me questions. I'd rather be interrupted than have a monologue. Okay, so right, so we talked about hybrid populations and this massive amount of genetic variation that's contained in them that can sometimes lead to adaptive evolution. But you know, it's a hard job to do genomics on hybrid populations. So for this project, we decided to zoom in just a little bit and we made a cross between two strains of the same species, Saccharomyces cerevisiae, which I think is a similar approach to what you often do in Drosophila. So here we can also ask more specific questions. In this case, we wanted to know how important standing genetic variation versus de novo mutations are for adaptations to new environments. And we also wanted to know how parallel evolution is from these two different sources. Right? These are both quite open questions that we don't know very much about. You're all familiar, I think, with this incredible long-term evolution experiment with E. coli that was started by Rich Lenski in the 1980s, I think, maybe 1970s, I don't know. This evolution experiment has or will probably reach 80,000 generations by this year. We've learned a lot about phenotypic and genomic dynamics from this work, but they were all clonal populations to begin with, right? So everything that happens in the system is due to de novo mutations. So we thought maybe we can do the same with yeast, but start with some standing genetic variation. So what happens to these evolutionary dynamics and what happens to parallelism if we allow for some standing genetic variation to begin with? So this is an experiment that was led by PhD student Kieran and a postdoc Lorena, who came in and did an awesome job on the bioinformatics. And what we did here is that we evolved a population that came with some standing genetic variation, which we created by crossing together two strains of Saccharomyces cerevisiae. And we evolved this population in replicates in 4 different environments. So these environments were salt, ethanol, and 2 different concentrations of lithium acetate. I always get the question, why that? Why those environments? They're entirely random. They don't really have an ecological relevance to it. We just wanted something new and something stressful, whatever stressful means. Okay, and we let them evolve in those environments for up to 1,000 generations. This is all asexual reproduction, right? There's no sex here, unfortunately. So let's first look at fitness. What happened to the phenotype? What happened to fitness? So we saw that populations increased in fitness, not surprisingly. This is a rule of thumb. This would happen in every evolution experiment. But they improved in fitness quite rapidly and quite early on in this experiment. So you see that in the first 100 generations or so, there was a rapid increase, and then it kind of just plateaued off. Not in every environment, right? There are some environments that where you didn't see really large changes in fitness. So, this is kind of consistent with declining adaptability or this kind of diminishing returns concept that beneficial variants have smaller effects in fitter genomic backgrounds. This is also something that, coming out of your group. So parallelism may be due to genetic redundancy. So there is quite a lot of parallelism in the system. I think especially here, for instance, or here you see that all 4 replicates evolved in a very similar way. So we think this might be because different subsets of small effect loci contribute to adaptation, but then lead to the same phenotype. Now let's look at the fate of standing genetic variation over the course of this experiment. So we selected about 10,000 SNPs that were segregating in the system that were shared by all the populations. And we saw, this is also something I've talked about today with some of you. I think you're looking into this in a lot more depth than we did. But many SNPs had shared allele frequency trajectories, probably due to genetic draft hitchhiking. So we reduced these SNPs to sets of correlated clusters, which is a beautiful paper also coming out of here. So we reduced them, and so what I'm going to show you next are basically haplotype frequencies. So this is an example plot. This is one of the ethanol populations, one of the replicate populations evolved in ethanol. And what you see here is that we call them jellyfish plots because they look like jellyfish swimming in the ocean. If a single diploid haplotype goes to fixation, many of the SNPs will fix here at the frequency of 0.5, right? These are sites that went to fixation and sites that went extinct. Now, I'm going to show you a lot of data in a minute, but I'll give you some time to stare at it. These are, again, the generations from 0 to 1,000, and these are haplotype frequencies. I'm going to show you the 4 different environments in these rows here and the different replicates in columns. And what we found is that environments actually differed quite a lot in these kind of standing genetic variation trajectories, or how standing genetic variation was sorted over time. Some populations fixed all of the ancestral variation very, very quickly, whereas others maintained variation until the very end. So here are some of these kind of largest differences that we think we identified. So in this strong lithium acetate environment, what we found is that a single dominant haplogenotype went to fixation. So this could be a haploid, or it could be an entirely homozygous diploid in the system here. So except for one replicate where there was a heterozygous haplotype. In other environments, we found a fixation of a single diploid here. This is kind of like similar to the plot I showed you before. So there is, lots of SNPs that went to fixation at this 0.5 frequency. But in other environments, especially in the salt environment down here, we found that genetic variation was maintained until the very end of the experiment. And this is the same case up here for ethanol. So yeah, so this is interesting, right? So it seems that environments differ very much in how much genetic variation is contained and maintained in them. And one possible explanation, we think, is that these dynamics are probably shaped by different genetic trait architectures. So it's possible that the salt environment has a lot more genes involved with small effect than the lithium acetate environment. Okay, so how about de novo mutations? Because we really wanted to know how different are the roles for adaptation of standing genetic variation versus de novo mutations in the system. So we also looked for de novo mutations. We identified about 300 of them. We kind of arbitrarily set the limit to de novo mutations that changed allele frequencies by more than 35%. That's quite a drastic increase, which might indicate that they have some adaptive value. Then we wanted to know, what are the dynamics of these de novo mutations? Is there actually parallelism at the gene level? So are genes or pathways hit repeatedly in different environments, different replicates of the same environment? That's what we wanted to look at. And this is showing you the same setup again, with environments in rows and replicates in columns here. And we found that parallelism at the gene level is actually quite high. So there were particularly 5 genes that have de novo mutations in multiple replicates. So for instance, look at the blue mutation here. This is ISW2. You find there's a mutation in that gene in several different replicates of the same environment, but also across different environments. Other mutations that were hit repeatedly were kind of private, specific to some of these environments. So the red one here or the green one up here were only found in single environments, but several replicates, which also indicates that they do have something to do with adaptation. Many of these genes were actually involved in a pathway that allows for the constant expression of genes that are involved in tolerating osmotic stress. Okay, but what I think is kind of cool here is now we can plot these de novo mutations on top of the standing genetic variation in the system. So every black line here is one of these de novo mutations that I showed you earlier. And you see that some of these drastic frequency changes in standing genetic variation actually coincides with the rise and the fall of some of these de novo mutations. So here's an example. Here, this mutation here kind of tracks exactly some of the haplotype frequencies from pre-existing variation in the system. Just a quick slide on this, because I also wanted to talk about one of these things that one of these questions that's been around for a long time, which is that environments with stronger selection should result in more parallelism. There is very little evidence for that. It kind of makes sense, but we don't have much data on this yet, and we wanted to look at this. So we saw that at least 3 of our environments kind of followed this nice gradient in stressfulness, if you want to call it that. So this is the fitness cost of these particular environments. And you see that ethanol didn't actually cause very much fitness loss in the ancestor, whereas lithium acetate 1 and 2 had larger fitness costs. So we have this kind of like stress gradient in our system here. And I think the answer to that is yes, tentatively at least. So here, these are the number of windows that are shared across replicates. And so the speed at which they fix and the number of these sites also increased with the stress level of the environment. So this is a bit complicated to look at. This is 100 generations and 700 generations, right? You see there's an increase. And the more stressful the environment was, the more parallelism in number of shared sites you can see. And you can see also that the lithium acetate environment, that was the one where the single haplotype went to fixation really quickly, already has strong parallelism after just 100 generations. And yeah, so these, we think we can answer yes, at least tentatively. What's that? Yeah, so salt. So I didn't plot salt here because it kind of, in terms of stress, it kind of falls right onto this one here, more or less. And there was less parallelism there than in the other environments that we saw. So salt is a bit of an outlier in that. It has all this maintenance of genetic variation. Yeah, so it's a bit different than the other 3. Yeah. So, conclusion, it's a little bit silly because it's just that everything matters. So we think that adaptation dynamics are affected by this intricate interplay of both standing genetic variation, de novo mutations, and also the level of stress that populations experience, but also the genetic trait architecture of resistance to these stressors. Right, so this is the last story I'll tell you. Bear with me for just a moment longer. This is data that we just got in. This is data by my PhD student, Alex Rike. And here we wanted to know whether we can predict evolution in complex environments from the effects of single components. And for this, we use clonal populations because it's a complicated question and we needed to make things a little bit easier for ourselves. So we did what almost every other microbial or yeast experimental evolution lab does. There are some exceptions, but we start out with the clonal population so that we can track de novo mutations over time easily. So we've learned from Peter and Rosemary Grant and many other people that populations evolve in the context of their environment. So this species of finch living on the Galapagos Islands normally eats these small seeds here. But then there was a drought in 1977, which caused all the little seeds to disappear. And instead, these large kind of spiky, hard seeds became abundant. And then a decade later or so, an El Nino event happened and the little seeds came back. And Peter and Rosemary Grant showed that beak evolution kind of exactly tracks these changes in seed and climate. But environments are complicated, right? Environments they consist of many, many different dimensions. We don't know if they all matter, but it's difficult to tell, right? You can imagine lots of biotic and abiotic factors that are determining the life and fate of these organisms. So can we predict the evolution of stressor interactions and adaptive trade-offs at all? What are our chances here? So there are 2 problems with trying to do that. The first one is that environmental factors can actually interact with each other and change each other's effects. So if you think of, for instance, just habitat loss and climate change as two different factors, they can sum up to the when they both occur together simultaneously in the environment, they might have exactly additive effects, right? This is a naive assumption, perhaps. But it could also be that they interact with each other antagonistically, so that the sum of these two is less than the additive result. So they might actually just mitigate each other's effects. And they can also be synergistic, so that when the two of them co-occur together, their stressor effect might be much, much larger than their sum. The second problem is that co-occurring environmental factors can result in adaptive trade-offs. So remember these seeds from earlier. So there's a bird, a species of finch that's adapted to eating those, and another species that's adapted to eating these large seeds. And these beaks are specialized. So these are probably, ecological specializations. And we probably can assume that there is no beak size that's optimal for eating both seeds, large and small, at the same time. So if there was such an optimal phenotype that can do that as efficiently as the two specialists, it would reside right at where these two dashed lines meet. But what's probably more likely is that there is a different bird that can eat both seeds, but not as efficiently as any of the two specialists. So that would kind of fall in this area of this plot. So keep that in mind, because I'm going to show you some results from yeast in a minute. So Alex and Dragan, a postdoc in the lab, they set out and wanted to test how bad are we really at predicting these stressor interactions and trade-off effects. So they evolved yeast populations over 15 serial transfers in complex environments. And these complex environments were composed of 4 individual stressors. So he used salt, starvation, so little sugar, an antifungal, fluconazole, and high temperature. So these are the 4 components. And he put them together in a full factorial design. So he evolved populations in just single factors, but he also had 2-dimensional, 3-dimensional, and 4-dimensional environments. Does that make sense? Yes. So these are what the reduction in fitness looks like before evolution. So if you throw the populations in and you measure fitness, which is often optical density in our case, in these 4 separate single dimension environments, you'll see that salt, for instance, causes a large reduction in fitness, heat, a rather small reduction in fitness. If you look at these 2-dimensional environments here, or the 3-dimensional environments, or the 4-dimensional environment, there's only one of them by design, you see that they have different kind of stressor effects, right? So the question again is here, how good are we at predicting how these interactions will evolve over experimental evolution? So let's look at fitness first again. So this is just showing you data from the transfer number 7 to transfer 15. So evolution for just 8 serial transfers. And you see that the high complexity populations here, so these are the 1-dimensional, 2-dimensional, 3-dimensional, 4-dimensional environments, populations increased more rapidly in fitness in the high complexity environment. I know the graphs don't look like they differ so much, but the average selection coefficients in the higher complexity environments are higher. Again, salt is a weird one out. So salt is the only one that had a pretty large selection coefficient. But on average, higher complexity means a more rapid fitness increases. But fitness variation among replicates increased with complexity. So that means, so this is 1, 2, 3, and 4-dimensional environments here. And this is the variation between replicates in that environment. And you see that there's a positive correlation So all the simple environments are down here, all the complex environments are up there. So you find less parallelism, essentially, in more complex environments. I think that also kind of makes sense intuitively. So can we predict the evolution of stressor interactions? So if you look at the ancestral interactions, this is before evolution, you can look at these small interactions that are happening in the ancestors. So this red means antagonistic, blue means synergistic. So there are some that are significant. So for instance, salt with antifungal and heat has a slightly synergistic effect, whereas others have very small interactions, at least, before evolution. If you look at the evolved populations after 15 transfers, you find mainly large antagonistic interactions in these lines. So especially here in the most complex environment, you find very, very large antagonistic interactions. So I would say that, no, we're not actually very good at predicting how these interactions evolve and what their stressor effects are over the course of evolution. The second question was, can we predict the evolution of trade-offs? So remember the Finch example here, right? If you had one evolved line that sits right in that corner, that means that it's as good at both adaptations to these single factors as the two specialists are. Or are we more likely to find some kind of like generalist that's okay, but not as good as the two specialists at both of these things at the same time? So I'm going to show you just one example from adaptation to the antifungal and adaptation to salt. So these two dots are the two single factor evolved populations. So you see the blue one here just evolved to salt, sits up here, and this is the green one that has only been adapted to the antifungal. If you put them both together, you see that trade-offs exist. So this is the same setup as up here, right? So at transfer number 7, adaptation to salt and the antifungal at the same time kind of was here. Evolution does improve that a little bit, so it moves up into that upper right-hand corner. So it means it does get better, but it never reaches the same level as the two specialists at either of the, actually very close to the green one, but not as good as the blue one. We do see that adaptation can compensate these effects. So we did see often that at first it was really a negative trade-off. So this is transfer number 7. And at transfer 15, it moved further to the two specialists. So interestingly here again, if you look at different levels of biological organization, parallelism was a lot higher at the genic level. So we identified about 200 de novo mutations in total in these experiments, and up to 19 of them were shared across environments with the same stressor. So this means in one-dimensional environments, 2, 3, and 4 environments, dimensional environments. I can show you in a second how that looks. Many times these were genes in the pleiotropic drug response family and in genes that helped adapt to high temperatures. Apologize for the large table. We yet have to find a better way to represent these data. But these are the different genes that we identified de novo mutations in. And these are the environments with increasing complexity. So you go from single environments to complex environments here. And you see that, especially in those green and the red genes, we saw that every time an environment had the antifungal in it, so this is the case here, but also in the 2-dimensional, the 3-dimensional environment, and the 4-dimensional environment, that gene was hit by a de novo mutation. And it's the second most abundant mutations we found in these PUF3 genes, which is adaptation to heat. So this is a single environment. This is the 2-dimensional and 4-dimensional environments here. And this is the number of times different replicates had been hit with the same mutations. So the conclusions from this last story, at least tentatively, is that parallelism is quite high at the genic level between populations that share the same stressor, no matter whether you look at a simple or a more highly complex environment. Parallelism in fitness is higher in simple environments than in complex environments. And trade-offs in complex environments are common and are actually really hard to predict. So this is my last slide. Experimental evolution is quite fun, but I think it's really lacking realism. So we've got lots of work to do. So we should make experimental evolution more ecologically relevant, I think. We should try to include more standing genetic variation. Sex would be a great thing to add, which we're doing at the moment in my lab as well. We should maybe strive to use more complex environment than these very simple lab environments. And right now there's a big push to take yeast research back to where the wild yeasts are and collect strains and species from natural environments to not just be limited to these lab adapted or domesticated strains that most people are working with. And that's it. I thank you very much.